So I understand this is a panel discussion for the Breakthrough movie, and we're happy to discuss the movie. I think you've either seen it or you're going to see it. I'm not sure how it's set up for your meeting.
Yes, perfect. So we have some minutes to interact with you, and there are some questions that we are receiving during this period until the Mexican Society of Oncology has launched this effort to communicate your improvements and merits in immuno-oncology. So we have some questions, and please, Dr. Cabrera, Dr. Vargallo.
So, Dr. Allison, Dr. Padman, thank you so much. We have a really honor to have you here in Mexico in our National Congress. Those big things talking about this. So, I want to ask something very soft. When does this start? This starts with your marriage. This starts with some idea of immuno-oncology. Does it start with love or with science?
Well, it started a long time ago. I had been, working on checkpoint blockade for some time from the basic science side, trying to get it into the clinic. Dr. Sharma was a clinician and was involved in designing some of the trials and working with the companies to, do the initial work. And so we met in the course of that, I suppose.
Yeah, I think, it started as great scientific collaboration and trying to make sure that the science Jim did in the laboratory in terms of curing mice could be translated to curing people. So we took that data from the mice and wrote a clinical trial so that we can test the agents in the clinic. And that was the first clinical trial that started out showing the efficacy of immune checkpoint therapy. So that's how it all started. It really started more of a scientific collaboration first.
And then we started working together after that. Those things were kind of done separately and came together, and then we started working together. And, one thing.
And the rest is history.
Yes.
So I was asking you, after you win this award, what do you think? Maybe this investigation line is over? Maybe like game over? The prize have already arrived?
No, tip of the iceberg.
Yeah. No, we're, the best responses to checkpoint blockade now are combination of anti-CTLA-4 with anti-PD-1 and melanoma, where the durable response is about 50% or so, and the other cancers are lower, 20, 30%. In some cancers, glioblastoma, pancreatic, no responses at all. So we've got a lot of work to do. The good news is that I think we know what a good response looks like, and so we know we got a lot more to learn, but we know what pieces are there: T cells, myeloid cells. So it's a matter of looking at individual cancers, maybe even individual patients to some extent, and treating them with something and seeing what happens, and then adding what's missing. You know what I mean? If we need to change the myeloid signature, we try to change that. We need more killer cells. We try to get them. We need more T cells in. We try to do something to do that. So, anyway, we know what it looks like at the end. It's a matter of getting the components together.
Yeah, I think we still have a lot of work to do. Obviously, we're not curing 100% of our patients.
That should be our goal, to cure 100%.
Exactly. So we have more work to do, and in all different tumor types. I mean, we've made great strides in many, many tumor types, but there's still some that are not, it's not working as well. So we're doing a lot of translational research with the clinical trials and also a lot of basic mechanistic understandings of how to combine different therapeutic modalities.
Much for your time, and it's for us it's an honor that you have been connected with us, and I would like to put two things in the table. One, a little bit I don't know, of the life, for to say it in a way, and another in a scientific point. The first one is, what do you say to the young people that is trying to working hard and found barriers to their work and rejects for the work and no funding, no? What would be your advice for them? And the second is, what happened with breast cancer? You know, it's a huge amount of effort and huge responses in lung cancer, kidney cancer, melanoma, but what's happening in breast?
So on the first part, you know, we want to give a lot of encouragement to the young people out there who are working so hard. We know that the funding is not always easy, and the work is not always easy. You know, we get into science and medicine because it's a passion. It's something that drives us, something that we love doing. You know, we have to be prepared for the ups and the downs in science. Not every experiment will work, and not every grant will get funded, and not every paper will get published right away. So it's always, you know, trying very hard to do something that you love very much. So as long as you have the passion and the energy to keep, you know, pursuing your dreams, you should keep working at it. And hopefully you have great mentors to help support you during the times that you are, you know, not feeling so great about your work. Your mentors can help to uplift you and provide you with guidance and advice. And so, you know, you have to keep doing what you love. If science and medicine is what you love, then it's not something you're going to give up very easy. It's sort of like a calling. That's how my mentor Lloyd Old explained it. It's a calling, and when you're called to do it, you'll always want to do it. And then in terms of breast cancer, I want to say, you know, we're very happy to have seen that there was an FDA approval in breast cancer for PD-L1 positive triple negative breast cancer now has approval with the tislelizumab and chemotherapy combination. And so now we just have to do additional work to understand how to get approvals in the PD-L1 negative group. And that's also important for how you think of a lot of other tumors, because you have to convert those so-called cold tumors that have very few immune cells and are PD-L1 negative to become hot tumors that have a lot more infiltrating T cells and to become PD-L1 positive, hopefully. So, you know, that's still work that we're doing, not just for breast cancer, but other tumor types like that.
I have to wear this, right?
You're free to do it.
Well, I think that all your investigation is really impressive, and we are all very honored to have you here. Well, there in a distance. But,
we wish we were there with you.
you are here in our hearts. You are here. What I really want to know, it's all the new stuff, especially in pediatrics, you know, in leukemia, all about immunotherapy in leukemia. I think there is a lot of investigation still to do. So that's my goal now.
That's great. I think that's a wonderful goal. And a lot of the CAR T cells are showing promise in leukemia and hematologic malignancies. So hopefully too, we can get to combination therapies with some of the CAR T cells and immune checkpoint therapy. And yes, we have to do a lot more work in pediatric cancer. So I wholeheartedly support your work that you're doing.
Sabemos que cuando hay tratamientos nuevos o hay un giro muy importante en terapéuticas, sobre todo a nivel de oncología, existe cierta resistencia en tomar estas nuevas pautas. A lo largo de toda su investigación, ¿cuál ha sido la mayor barrera, independientemente de la económica, en cuanto a proceder con la investigación en inmunooncología?
Well, the first was just getting to do it at all because people were so skeptical. It took me 4 or 5 years of going to company after company after company to get people to even try it once, you know, even to try it at all. That was the big one. So now I think it's much easier. I think the stumbling blocks now are to more subtle, and to try to get people to do more science instead of and look at biopsies and look at changes that are occurring, rather than look just for a clinical signal. Mm-hmm. And it's slowing us down a lot, because a lot of patients are being wasted by just, you know, treat with a combination. And if it doesn't work, you don't get a signal, drop it. But we know that some things may happen that may inform what to do the next time. Mm-hmm. And it's frustrating ever to treat a patient and not learn something.
Yeah, I think the major obstacles that we're dealing with right now are how to have rational combination strategies. I think the first hurdle that Jim had to face was really getting immuno-oncology started, to show that it can make a difference in patients' lives and to get the first FDA approval. And that happened in 2011. So it's been 9 years, and now it's all about making sure we have the rational combination strategies, and we're understanding enough data from the human side so that we know what's happening in human immune responses, so that we can come up with the next pathways that need to be targeted. So I think that's what we're dealing with as a field. I think on an individual level, you know, certainly the Nobel Prize opened up a lot of doors and avenues for us to have a platform on which to stand and move the field forward because of that. But before that happened, you know, on an individual level, it was convincing a lot of the other clinicians or researchers that this was a worthwhile path to follow. So it was a lot of hard work to try and get those first few clinical trials started and to get patients accrued on them.
Okay, so Dr. Bargallo.
La inmuno-oncología debe de empezarse a enseñar ahora a las nuevas generaciones, tan temprano como desde la medicina,
yeah.
o tan temprano como desde high school.
As early as learning chemotherapy immunology should be taught. Immunology should be taught when you learn about biology, about the blood system, any other, when you learn about the body and how it works.
I think in college, right, in undergraduate classes, when you're taking basic chemistry or basic biology, there should be a course in basic immunology. And then when you get to medical school, clearly there should be a class on, you know, immuno-oncology, the same way that, you know, they teach a lot of other courses in medical school. And the earlier the better, because then I think we'll have more experienced physicians who can give immunotherapy to patients.
Bit complicated being a low middle income country and look out all the trials and the combinations and the magnificent results, and get to the clinics and guess that your patients can't not afford those drugs. So what are you thinking about from the leadership of a Nobel Prize? Could you are thinking or do something for the access to the rest of the world?
So yeah, we serve on a lot of committees, and you know, we have conversations with a lot of different pharma partners about the idea that, you know, drug accessibility is an issue. I mean, we don't want it to be that you have to go to a premier medical center in the United States before you have access to drugs. But it's also an issue with every country having its own health insurance system, and how the drugs can be provided in a way. How do the companies get paid? And every country is dealing with a different issue. I do know that some countries are trying to produce the drugs, so-called biosimilars. So it's not exactly using the same drug from Bristol Myers Squibb, for example, or from Merck, or from, you know, Genentech. It's getting a biosimilar to CTLA-4 blockade, or PD-1 or PD-L1 blockade. So they're making their own drugs within the countries. That may help also in terms of access. I do think these things need to be addressed, not just from a scientific perspective, but also government agencies have to be involved, and patient advocacy agencies have to be involved.
We'd like to see it democratized, so that as many people as can get it as possible to the whole world.
Yeah. Cancer care should be equitable and everybody should have access to appropriate cancer care.
I would like to know your opinion about the interaction between immunotherapy and radiation therapy.
Now, there's a lot of work suggesting it can be very good. The thing though is that you have to change the way that you do radiation therapy. If it's done in a fractionated way where you kill cells and the immune system comes in, to deal with the inflammation and start priming T cells, then you irradiate again at that point and kill everybody, then that doesn't work so well. And so there are a number of groups now that are trying to adapt radiation. The goal of radiation therapy has always been kill every tumor cell, everyone, because that's the only way it works, really. But now it's just kill enough to get the immune system to finish it off, and don't hurt the immune system any more than you have to in the middle, And so there's a lot to be done, but I think there's a lot of promise there.
Yeah, I think the radiation therapy can be used to prime the immune response, to possibly make a cold tumor hot by allowing for some tumor cell death, and then antigen presentation with dendritic cells and macrophages. But the radiation therapy can't be given in a way where it's so high or so frequent that it's killing the macrophages or the antigen presenting cells. So, it's really trying to come up with new ways to give radiation therapy so that it can synergize with the immunotherapy agents.
Doctora Rubalcaba.
¿En algún futuro la inmunoterapia sustituya a la quimioterapia?
No, we think that actually, that immunotherapy can be combined with chemotherapy in a way that you don't have to give so much of the chemotherapy, which will be good because then you won't have the toxicities of chemotherapy as much. And also in certain setting, maybe we can give immunotherapy up front in earlier disease stages. And then if need be, you can also give chemotherapy at that time. But the point is to get the memory response of the immune system to prevent recurrence of disease or to prevent relapse of disease. So either it'll be immunotherapy or immunotherapy plus chemotherapy, but each disease type and each stage of disease may require different treatment.
I think chemotherapy as a treatment by itself will disappear. But I think that one of the things that when I first thought of this in the 90s was the whole process starts by priming T cells because of tumor death and inflammation. And to the extent that you can get chemo to do that, if you've got a tumor that responds to a certain chemo, then maybe you can drop the dose of the chemo so that it's less toxic, doesn't hurt the immune system, just kill it. Again, the idea, just kill enough to get the immune system come in and provide the benefits that it can. But that's another way of priming more efficiently. If you just give these immunotherapies by themselves, you're relying on the tumor cells to just die of their own accord or something. That's what starts the process. But if you can accelerate that and know when it's happening, even more important, with radiation or chemotherapy, then that's sort of your priming dose. That's an initial vaccine. And then you build on that. And I don't think chemo is going to go away. But it's got to be changed fundamentally to where it's used not, again, to kill every tumor cell, not to cause all the toxicity that it does, but just to kill enough and cause enough inflammation to alert the immune system to go.
I mean, in certain diseases, chemotherapy works very well. If you think of germ cell tumors, for example, so you wouldn't want to get rid of chemotherapy when it works that well.
Certain childhood leukemias.
Yes, and another question is, I mean, your focus is in immuno-oncology, but do you have any research or any other steps in other kind of treatments that maybe in the near future we can discuss as a new Nobel Prize with discoveries or something? Or maybe combining therapies, or what do you think about the next steps or the next future in oncology?
I think the next future is definitely still going to have immunotherapy involved. It's just going to be what are you combining with immunotherapy. So one of the things to consider may be epigenetic modifiers, so that you can epigenetically not only alter tumor cells, but epigenetically alter the immune cells, so that maybe you can get better T cells, or you can get better myeloid cells or better dendritic cells to work within the immune response. I think the agents that people are looking at for epigenetic modifications are to be taken into consideration. And obviously, I think the space of engineering is leading to better protein engineering, so that it's no longer just CAR T cells, but bispecifics in a way that can target different, whether it's a tumor antigen and a T cell at the same time, to allow for this bringing of the T cell closer to where the antigen is expressed. I think these are kinds of things that we will see coming in the future.
About the combination with the immunotherapy and oncolytic virus, there are the possibility for this combination?
For the combination with what? I'm sorry, immunotherapy and?
Oncolytic viruses?
Yes, oncolytic viruses. Absolutely. I think there are clinical trials now looking at that. It's going to be a matter of which oncolytic virus, because they're different backbone, whether it's an adenoviral backbone or Newcastle disease virus backbone, poliovirus backbone. These are things to consider. TVEC was FDA approved, the Amgen drug. So it is possible, of course, to have the viral treatments. Not only which backbone, but how to give it, whether it's going to be intratumoral or intravenous. These are all kinds of studies that are ongoing right now. But I think the oncolytic virus has a great opportunity to prime also an immune response, and also to drive an immune response against the viral components, which hopefully will have some additional cross-reactivities with the tumor antigen expression.
The virus approach offers the ability not only to kill the tumor cells perhaps in a much cleaner way than radiation or chemotherapy, but you can package certain genes into the virus so that you can induce immunostimulatory molecules into the tumor cell as it's dying, to further increase the ability of the immune system to see it. So the ability to engineer oncolytic viruses is huge, and I think that's going to be a very, very important area. We've been working a little bit on that in my laboratory.
That's very nice. So I have a question from Monterrey in Mexico country. So,
buenas noches. En nuestra ciudad, Monterrey, iban a estar ustedes invitados al Congreso. Esperemos tenerlos muy pronto. Muchas felicidades. ¿Ustedes sienten que ya le entendieron a la inmunoterapia o están rascando la superficie apenas?
Well, we're beginning to understand it.
We're scratching the surface.
Imagine us. Y la otra pregunta, en nuestros países donde el acceso es un problema, ¿sería relevante conducir estudios clínicos buscando dosis mucho menores de inmunoterapia combinadas con otras formas de tratamiento? ¿Y habría modo o ideas para conseguir el funding, que obviamente no vendría probablemente de la industria, sino de otros fondos independientes, para conducir investigación con dosis más accesibles de medicamento?
So I think you're absolutely right. One, we have to try and figure out different doses and schedule, because sometimes the toxicity is too much at certain doses. And so if you notice, some of the approvals, for example, with anti-CTLA-4 or Yervoy or ipilimumab, is approved at 1 milligram per kilogram. Some are approved at 3 milligrams per kilogram. So this is something that has to be looked at depending on the tumor type and maybe the patient population, as well as if it's being combined with something else. And then two, accessibility in countries that may not have the pharma partnerships in order to do the clinical trials. That's something that we have to bring together at a bigger oncology symposium. We have the ASCO symposium or AACR symposium or Society for Immunotherapy of Cancer symposium. These are international groups that we need to bring together, that we discuss this. as a body on how to make sure that we can open up the clinical trials at other sites. So even though they're being opened in the United States, to have them opened at other places with funding intact from the organizing bodies. So that's something we're trying to see how to mobilize it. I think COVID also threw a wrench, a problem into it, because with the COVID shutdown, all clinical trials were shut down, even the ones in the United States. So we're just starting to get those up and running again.
Thank you.
So thank you for the final part of this meeting. I really appreciate, and I'm sure that our associates love all your thoughts and talks about the immunotherapy. At the final, I ask to you, both of you, that a final message for our people in Mexico, our oncologists in Mexico that are still fighting with melanoma, lung cancer, genitourinary cancer, all the cancers, and maybe receive your words, maybe convert to superheroes like you.
Well, we wish everyone luck and, happiness and success in all of your work, because really we all have to do this work together. We don't see this as just the two of us. We really work with large teams all over the world. So, we hope you all enjoy your meeting and that you come up with new treatments that you can teach us about at some point.
Dr. Allison?
Yes, thank you for giving us the opportunity to meet you, and maybe we'll have a chance to meet in person one day.
Thank you.
So thank you. Receive our gratitude and admiration and a big applause from Mexico to Houston.
Thank you.
Thank you.
Thank you all. Bye-bye.
Bye. Ahora podemos pasar a que ustedes conozcan la historia, y realmente es un cortometraje sumamente emotivo, sumamente interesante para quienes somos médicos, quienes somos oncólogos. Lo van a vivir de una manera sumamente intensa. Los dejamos con ellas y al final regresamos con ustedes. Muchas gracias.
It's a part of Texas that was still majority Hispanic. Football reigned, everybody, Friday nights, that was what it was all about. And it was an oil boom town too, although there was a lot of agriculture as well. My mother's family's been there since probably the 1840s, I think. My dad was a doctor who, took care of people in the neighborhood, did house calls. My father moved to Dallas in the 30s sometime. I don't remember exactly when, but I was born in 1948, and we lived, I think, here, actually, because there was a washing machine out in the yard, and I got my arm caught in the ringer one time. It pulled me up and, got stuck, and I was hanging, by my arm, and some neighbors came out. I have two brothers, Murphy and Mike.
My mother, she was a housewife. She was a good influence on us, just kind of give us a little discipline. We get to wash the dishes on Sunday and things like that. Of course, that may have been a little bit before Jim's time. I think he got out of that.
I didn't really know what was going on in those days. Nobody talked about it. All I knew, she was sick, and she had these burns on her throat, and she was in bed a lot. I remember one day had some good friends, had a kid my age, and his mother was going to take us to the swimming pool. I'm just about to get in the car, and somebody came running out and said, "Hey, you got to come back. You can't go," you know. So I went in, and she was dying, and that made her. So she died that morning. And I found out later, I really didn't know what it was, but I found out later that it was lymphoma. She'd been getting radiation therapy, hence the burns on her neck. I sat on the bed, you know, with her and held her hand while she died. You know, there was certainly a big hole, a lot of loneliness. I was 11 years old when my mom died. My father was just heartbroken. You know, he seemed to kind of lose interest in things. Anyway, he was gone a lot. I ended up living with sort of a foster home. I mean, not in the sense that I was giving away everything, but a counselor from high school teamed me up with a family who had a boy about my age, and so I spent when my dad was away. I stayed with them all the time. I like to wander around in the woods. I lived right at the edge of town. I used to wander in the woods by my home. I just found it was comforting to have a harmonica and just toot away, you know.
On behalf of the Nobel Assembly at Karolinska Institute, I wish to convey to you our warmest congratulations. May I now ask you to step forward to receive the Nobel Prize from the hands of His Majesty the King.
Innovation is extremely difficult.
We were all very skeptical.
Many people said, "Jim Allison, you're wasting your time."
I knew that this is a way to cure cancer. I knew that that was a way. And I'll be damned if I'm going to let that just lay around. Well, I guess my dad encouraged me to mess around with my Gilbert chemistry set in the garage, you know, and dissect frogs and horned toads and things.
He told me about the bombs he used to make. So he would construct these little bombs out in the woods, and he said, in this very Jim way, he's, how, when you talk with scientists, later in life, you realize a lot of us were bomb makers in the beginning.
When I started learning about what science really was and how to do it, I started doing more intelligent things than making bombs. One of my teachers who taught advanced mathematics, physics, chemistry, I thought he was one of the greatest teachers I ever had. But I tried to have a few conversations with him about evolution, selection of the fittest, Darwin. And he just said, no, it's not real. I do not allow. We will not teach that here. We will not discuss it. I refused to take the course because I knew that there was no biology in it. The syllabus, there was no biology in it. So it was a pretty ugly time. And it was basically me versus him. I was still taking advanced physics from him while I was having this fight with the school because he was the head of the science program. He was the one that made sure that evolution wasn't taught. And so he really did start taking it out on me. back in those days, you could use boards to paddle students, so I seemed to be getting in trouble all the time and getting a couple of licks just for walking by, They just thought I was a troublemaker. I wasn't. I was just trying to do what was right. That's the way it is, If you don't agree with something, you need to stand your ground. And if it hurts, it hurts, It's the way it is,
When he was little, we said he was hard-headed. We never did call Jim this to his face, but, this guy, we called him Diamond Head, because his head was the hardest substance known to man.
Jim graduates from high school at age 16. He goes to the University of Texas in Austin, determined to become a biologist.
There was this knock on the door. Door opens and this scrawny kid pokes his head in and says, "You got a job?" I said, "There's one job left, but you may not like it. It's just a dishwasher." And he said, "I'll take it." We would take a piece of cheesecloth and put a little bit of acetone on it and rub off the number of each test tube. And literally there were thousands of them. And so he got the idea, well, maybe I could take those test tubes and spray them with a solid stream of acetone. This was back in the 60s, and just about everybody smoked then, including in the lab. Jim sprayed his test tubes with acetone and then sat back to have a cigarette. And boom! And we all run in from the lab, and here's Jim with a blackened face, and his eyebrows were all singed. I was about to fire him, but in the short time he'd been there, Jim had just got along and knew all the graduate students and the others, and they besieged me on his behalf. Look, can you find something else for Jim to do? He's quite a character. Let's keep him around.
Well, I'm a king there. Want you to be my queen.
In the summer of 1966, he came for a fraternity party that I went to with someone else.
Together we can make honey, baby, that the world has never seen.
He was the most amazing thing I'd ever seen in my life. I never dated anyone else in college. We were together from then on. It was a really kind of magical time to be in college. It was so much fun, so much going on.
We were lucky enough to be in college in Austin just as everything was beginning to happen there. That really made it when Willie Nelson moved there. I think the first time I saw him live was playing on a flatbed truck at a Ford dealership on South Lamar in Austin.
But everyone was also real serious about their studies. Jim was passionate about science.
When I was at the University of Texas, the number one thing I learned in the lab was just how important it is just to persevere. Not be so disappointed you give up, just keep working.
We could never go anywhere without going by the lab, checking on this or that experimenting process.
T cells were discovered when I was in college. And I took an undergraduate class, and my professor, Bill Mandy, taught one lecture about T cells because almost nothing was known about them.
The human immune system circulates in our blood and protects us from disease. We now know that at the center of the immune system is the T cell.
T cells are a type of white blood cell. And these are the cells that are actually responding as viral infections, bacterial infections, and they start the fight to keep you healthy.
These cells that go all over your body and manage to protect you and not kill you. It's still, to me, just wondrous. I wanted to try to do something that Barry Kiddo always taught was, you should try to do things. If you're going to do this stuff, you ought to at least do things that help people. My mother had 3 brothers. One died from melanoma. In those days, melanoma was a death sentence, so he didn't even get therapy. When he was diagnosed, he just said, that's it. Had a really good time for the months that he had. The other brother was a former rancher who could roll a bull-durham cigarette with one hand while he was on a horse, but he paid for that. He had lung cancer and died from that. I saw him in an oxygen tent when he got to the point that his emphysema, whatever, was so bad, he needed help to breathe. Those made kind of an impression.
My own lab was not involved with cancer when Jim came to work with us. But he had had a tremendously difficult relationship with cancer. So at graduate school, he convinced me that perhaps we could look at something in the cancer field.
There was an enzyme called asparaginase that, when injected into kids with leukemia, could induce remissions. I was first thought they were cured, but it turned out they were. They were for a time, but then they always came back. My thesis project was to find other asparaginases from other bacteria that would be so different that they could be used in succession.
We pulled water out of the local streams and surveyed that for asparaginase activity. Jim purified that. And in mice, that worked very well indeed.
Jim's work on asparaginase convinces him the immune system plays an important role in responding to cancer.
I was a bigger kid growing up. I really ate way too much sugar. I know that now. I played a lot of sports. Basketball was my thing. I played my first year of high school and then injured my knee pretty darn bad. Kind of lucky that I did, because I couldn't play basketball anymore, but I joined the marching band, where I ended up meeting my soon-to-be husband. I was finishing up my graduate year. I had what, feels like bronchitis. You were coughing, breathing was a little bit of an issue. Progressively over the next couple of months, I didn't get better. I was walking up a hill one day and I could not take a deep breath. My mother worked for a primary care doctor, and she's like, "Oh, sweetie, when you get home, come on, see him, and we'll get this sorted out." Little did I know that just a few weeks before that, I was going to develop a mass growing out of my chest, and I thought it was breast cancer because of where it's located. So going to see her doctor, They noticed the tumor and they did a biopsy, and my world changed.
I went to Scripps because I wanted to learn about the immune system. I wanted to ask questions about how the immune system worked.
Strangely enough, there was a group of expatriate Texans there, and we would all go to a little bar called the Stingery. And there was a band there that he would sometimes play harmonica with.
Columbia Records was having their annual convention in San Diego, and Willie Nelson's record, The Red-Headed Stranger, had just gone platinum, so they decided to have a big party for him. The guy that was in the sheriff's department, was a real devotee of our band, came and told me about this, and said that we're doing security for it. He let me go in, let me through security to go to this private party. Willie Nelson came over and he said, well, you know someplace we could go picking tomorrow night? Because I got tomorrow night off, and I'd sure like to play some music. So I said, well, it's talent night at the Stingery.
So we loaded up in our little VW camper. We walked in, and people just fell on the floor.
Willie basically asked, you guys mind if I play? He had his drummer and his bass player and started playing. They played for 3 hours at least. And, it was a little dive bar, and there's about 8 people at this place, There was one payphone on the wall, and so those 8 or so went up and called people, and pretty soon the place filled up, and people called other people, and it was full, and then people were out on the street. It was amazing. I didn't have to buy a beer at that place for about 2 years after that. That old college roommate of mine, he heard that MD Anderson was opening a new laboratory near Austin. To tell you the truth, the main reason I went there is because I thought I would have freedom.
The first time I connected with Jim was, I think, in about 1980. I was a postdoc in New Mexico. I went to MD Anderson, and they asked me to give a seminar. And they said, well, you're being live streamed to Smithville, Texas. And so I gave my seminar, and they said, do we have any questions from Smithville? And I'm thinking, where in the hell is Smithville? And then this voice comes over with this, nice Texas accent and asks me this incredible question.
With a given molecule on a pathway, for example, the MAP kinase pathway, which, is
He
involved in
got right to the heart of, the hardest question he could ask about the science I was doing at the time. I consider Jim my brother, scientifically as well as personally. I would consider him my best friend for the last 30-odd years.
I really wanted to understand the immune system and everything, so I really wanted to know how T cells worked. And at the time, all we really knew was that T cells went around the body and took care of stuff and didn't hurt you.
Scientists know that T cells distinguish between healthy cells and threatening ones by using a molecule on their surface called the T cell receptor. But they don't know how the receptor works.
We didn't know how those white blood cells, these T cells, would actually recognize the virus as being different and needing to be eliminated. And that was the holy grail in the late 70s and early 80s. All the top labs in immunology, that was the quest.
I read the literature, and there was a lot of stuff that was contradictory, a lot of stuff that seemed like fantasy. I could not possibly imagine how some of the mechanisms that were proposed actually worked. I said, well, either I'm a total moron and can't follow this complicated stuff, or people are just, it's all nonsense. So finally I said, forget it. I'm not going to pay any attention to the literature.
Part of Jim's success is the fact that he's an iconoclast. You know, he doesn't care. He doesn't care that he's not following convention. He's following his own thoughts, his own motivations. He's willing to do it in the face of his colleagues not necessarily believing him.
You got to notice that Irv Weissman from Stanford, who was a very noted immunologist, was giving a lecture. So I drove 3 hours from Smithville into Houston. I was driving back, replaying his talk in my head. I remember it just sort of clicked, and I said, I know how to find the T-cell receptor.
Great scientists are creative people. They're not just focused on their detailed work. They're thinking beyond. There's a creativity to this work, and frankly, if not for the creative scientists, we would make little progress.
I saw miles and miles of Texas, all the stars up in the sky. I saw miles and miles of Texas.
Drove back that night and just rolling in my head and everything, and you know, the next few days, just lying down the path to find the T-cell receptor.
Over the next few months, Jim uses the latest genetic engineering techniques to test his hypothesis.
Work hard all day, get an incubation period or something, go to the beer hall, you know, knock down some beers, go back and finish the experiment, you know, go to sleep, get up, start it all over again.
I think the guy is just incredibly passionate. So he'll spend a night going out to a honky-tonk, and he'll be out all night, and he'll drink, and he'll have a great time. And then there's that same energy that would put him in the lab at Smithville. They're doing a 36-hour experiment, and he's staying up for that. And he's just as driven and turned on by that whole experience of being in the lab.
We went to a conference in California. This is where you present your data. You're not well known enough to be invited to get up on the podium and give a talk at that point. You know, you're relegated to putting your work on a poster board. So Jim had a poster there, and I remember walking over to look at his poster, and we'd just kind of met recently then, and he pointed to this spot on the poster and says, "That's the T-cell receptor." And he was right.
His contributions to our understanding of the T-cell receptor, the molecule most centrally identified as the thing that recognizes the target and allows for it to be eliminated. Extremely important.
He was the first one to actually purify the molecule by which T cells recognize everything. This is how T cells recognize infectious disease. This is how T cells are recognizing things they shouldn't recognize when you get autoimmune diseases. So this was the seminal discovery.
Jim was invited to major immunology conferences, and it was recognized that this constituted a real breakthrough.
Guys. When they came back and said, "Sharon, you have melanoma," I was completely shaken. Unbelievable. I couldn't believe that that was my life. I thought I was going to die.
Good job.
The doctor canceled all his patients and shut his office door and got on the phone and said, "I have a 22-year-old patient here who needs to see you now." And he was on the phone all day. but I had an appointment with Sloan by the end of it. So then I saw Dr. Walchuk just a couple days later.
Sharon Belvin, when I first met her, was 23 years old. She was seen by an oncologist near her home in the New Jersey Shore, who called me and said, "You need to see this young woman." So I see her, and it's a room full of very sad, family members trying to get their head around this seemingly unbelievable situation. We started her on chemotherapy very quickly thereafter.
So I had chemotherapy Monday, Tuesday, Wednesday, and got married that Saturday. We decided to go through the wedding because it's for better or worse. I mean, we were joking around a couple days before, and we were just like, okay, well, we are going to get the worst over with right in the beginning, and then the rest of life will be great. And Rob is really probably a saint, and don't tell him I said that. Who can look at somebody and say, here's a terminal diagnosis, and I'm going to marry you anyway?
In 1981, there was a bill that was introduced by a very conservative state senator from East Texas saying that creationism needed to be taught in schools.
One of his teachers from Alice had actually been elected as the state representative from Alice, not the science teacher, of course. And she called him up and said, someone has introduced a bill to outlaw the teaching of evolution in the public schools, and we've got a hearing coming up, and we need someone to come and talk.
She said, he's going to have 300 people here supporting the bill and opposing evolution. And so if we don't have somebody here speaking to the other side, it's likely to pass from committee out to the House floor, and God knows what's going to happen. I tried to find other people who would do it. I only found one other scientist that was willing to do that.
I'm here tonight speaking before this committee because I feel that in this highly technological age, an education in science is essential if members of our society are to be able to understand and deal with complex and diverse problems facing them. And I know from my
I said, science is something that you can use. It's not just accounts for the past. It does that. It also allows you to understand what's going to happen in the future, to situations that you've never seen before, that have never arisen. That's what science is.
That is the chief problem with creationism, purporting to be a science. It cannot be used to construct explanations of newly observed fact. It does not give us any help whatsoever in understanding how bacteria acquire resistance, how tumor cells arise, or how they are eliminated by the immune system.
He talks about using the immune system to cure cancer at that point, even in 1981.
I said, I challenge this man to use his creation science, such as it is, to understand how a tumor escapes the immune system. It's hilarious, the limits of their arguments. Probably the best one was, if you put a Ford in a field, it rusts, it don't turn into no Cadillac. I said, oh, okay, I got it. Obviously, it can't be anything like evolution.
Pretty soon after that, he got an offer from the University of California at Berkeley to go out there.
After the T-cell receptor, I was offered a full professorship at Berkeley, and I was really happy out at Smithfield, living in Austin, working in Smithfield, just doing science. It's as clean as it could get. I was doing exactly what I wanted to. I wasn't answering to anybody. I didn't have to do any administration or anything. And so the idea of going into some big-time place and having to, compete was a little scary. But an old friend of mine called me up and said, what are you doing And I told him that I'm comfortable here. I'm happy here. He says, yeah, in 20 years, they're going to be sitting there with their feet up on the porch saying, I could have been a contender. I kind of went, yeah, I understand what you mean. When I decide who to hire in my lab, I always am looking for one thing, really, and that's curiosity.
Berkeley drew me in a lot of different ways. It's filled with very smart people, but very freaky people, and they all sort of intermingle, and you don't ever know what's a crazy idea and what's a great idea. Even at Berkeley, Jim was a standout. He didn't look like the other people. Berkeley has its own conservative liberal side of it, which is that, you know, you can be liberal up to a point, but Texan is actually too far. You know, it's like you're carrying a gun and I don't know, all kinds of things that could be bad. And so Jim was like just over that line and represented just the, you know, additional frontier even of Berkeley. But also, immunology was not considered a science. The classic people said, oh, immunology, that's sort of a pseudo-discipline. That's really like almost like giving holistic medicine or something, right? It's like, what is that? You don't even know what that is. So Jim's lab has a feel of being like a pirate ship. There are personalities doing their own crazy stuff, and yet there is a, you know, dedication to the purpose of driving the boat forward.
I was working a lot. Not as hard as the people in the lab, of course. I mean, they were working all the time. But we also all partied together. You know, I just had some incredible parties.
There is the rigid thought process that is like connect the dots, A to B to C to, you know, like that. And that you do during the daytime. But I think there's a whole element of the idea of unwinding and not letting the mind go linear and just saying, what is, you know, what if it weren't, what if nothing we thought was right?
Jim's work on the T-cell receptor has sparked a race to discover more secrets of the T-cell. Soon a French scientist finds a new protein on the surface of T-cells called CTLA4. Most researchers assume that it activates the immune system like the T-cell receptor. But Jim doesn't assume anything.
The only right way to do science is not to do experiments that give you an answer that's consistent with what you think you ought to get, but do the killer experiment that can only be right if you're right.
Jim started looking at the data on CTLA4 critically, and he and Max Krummel, his graduate student, started saying, well, let's see what the real data is. If we do experiments, is that true or not?
So we just had this stupid question, like, what does it do? And it wasn't like just cure cancer. It's like, well, what's that do?
Are you shooting him?
Yeah.
Well, it's Christmas. It's 1991, and we're loading up the piñata here. See. Here's Robert.
Robert?
To study CTLA4 in a mouse, Krummel must build a mouse antibody, a Y-shaped protein that will trigger a reaction from CTLA4.
Yeah. Police car. Sam left Robert the police car.
Okay, let's go.
What made that whole 3-year stretch for me very difficult was that I didn't get any progress scientifically.
Robert's fourth birthday. Robert, look over here. Look, are you Batman?
And this is really common. For 3 years, nada. You know, like everything I did really didn't amount to anything.
running laboratories, it's not easy to have people do the experiments you want them to do. I regret at times not being able to motivate people to do the kinds of experiments that I was sure needed to be done. But Jim has that ability to do that. So because of that, people in this lab have done things that were crazy at some times, but he also was able to get people to do the things that he was really passionate about.
While Max struggles to make his antibody, another lab beats him to it. Peter Lindsley, a scientist working with the drug company Bristol Myers Squibb, publishes results saying that CTLA-4 is an activating receptor. When it's engaged, it revs up the immune response against targets.
Then these papers came out and said it's a positive regulator. And so, that attitude was, well, we've been scooped, it's done. Except I said, I don't think so. There's something about, I saw some holes in that paper, and so we're going to test this fucker and see if it's really right.
You have this picture of Einstein sitting at the whiteboard, hair frayed, writing equations that no one can see. Really what he's doing is saying there's a fundamental question, an assumption people have made that doesn't make sense if you look at this in a clear-eyed way. What if I step back here and just observe and say, these pieces of information need to be put together in a different way? I think that is what pioneers do, and weirdly, it's childlike.
This is a hard thing. It took me a while to be able to say this to people, but I think there was a formative moment when I was in Jim's office, and I actually started to cry. Because I'd been working on something for so long, and he told me something, again, a mistake that I'd made, and I just started to cry. And then I got over that. I mean, there's a period of that you just have to get through and just say, okay, go back at it.
Crummell finally makes his CTLA-4 antibody and launches Jim's experiments. He quickly demonstrates that Lindsley's idea of how CTLA-4 works is wrong. The experiment proves that engaging CTLA-4 doesn't activate the immune response, it stops it.
Jim and Max Crummell, as well as Jeff Bluestone and his grad students, turned the thing completely around, said, nope, all these prior papers saying that CTLA-4 is another gas pedal, that's not correct. It's actually a brake.
Like Jim, I was able to figure out CTLA-4, and I was shocked. CTLA-4 was the wackiest thing in the world, right? Nobody believed that you would actually have a whole system in place to shut down the most important thing going on in your body, which is an immune response, and you're going to shut this thing down and make sure it doesn't work.
The body does not want an over-exuberant immune system, because if we did have one, we would all be suffering from autoimmune disease.
Autoimmune diseases, like multiple sclerosis, are caused when the immune system attacks healthy cells. Jim's discovery prompts many immunologists to think about curing these diseases. They will engage CTLA-4 and stop T cells from going on the attack. Jim heads in a different direction.
There's this big part of the immune system that is built on stopping itself. Now, what the heck does that have to do with cancer?
Cancers, we know they have mutations. We know they're different from normal. They should be visible to the immune system. And for a long time, we struggled. Why can't the immune system see cancer? It can see flu.
Jim theorizes that tumors have evolved to trick the immune system. They engage CTLA-4, turning on the T cells brake. This stops T cells from attacking the tumors.
If you were a cancer, it would certainly be advantageous if you contacted that receptor and turned off all those T cells that might otherwise come to kill you. this would make sense. At the time, it was a crazy idea. Nobody thought tumors were trying to turn off the immune system. But he said, well, what if I block this? Why don't we see what that does to certain cancers?
Jim hypothesizes that if he inserts an antibody to block the connection between the tumor and CTLA-4, the T cell will be free to attack the tumor.
Jim had all these tumors in the freezer. So I was making antibodies as fast as I could, and a guy named Dana Leach came. He was a postdoc in the lab, new postdoc, and Jim gave him the tumor models, and I gave him the antibody.
The student who was doing the experiment had put the tumors in the mice and gone away for the holidays. And so it was actually left to Jim to check on these mice, and shortly before Christmas, I think, he saw the tumors are growing, maybe nothing's going on. Kind of sad about that. It's not a happy thing to see, but that's what the data was.
We had Thanksgiving and Christmas together for probably, almost 20 years. I remember that Christmas. He was mumbling about, the student taking off and going on holiday.
I had family visiting and everything, and so I did something that was unforgivable, really. If he had done it, I'd have fired him probably, but I skipped a couple of readings for the Christmas holidays.
A few days later, though, Jim remembered, oh yeah, I'm supposed to be checking on this guy's mice because he's still off celebrating Christmas with his family, and came back to find that, in the mice that had received the antibody, the tumors were gone.
It was one of these, truly one of these aha moments when I went, oh, Jesus, this is something big. Because I've been doing tumor immunology enough by then to know this experiment was so cool, you didn't need a statistician. They were all alive or all dead,
These tumors have disappeared. I mean, this is impossible. This is not possible. That's how mind-boggling you have to think of this as, because the human body, with 480 million years of evolution behind it, can't get rid of this stuff. And now the tumors have disappeared.
From then on, for the next decade, essentially he had one goal: to get that made into a drug for cancer patients.
It didn't come out of cancer, but once it was there, it was there. And I'll be damned if I'm going to let that just lay around. This is a way to cure cancer. I knew that was a way.
Well, I'm a kingpin, buzzing round your hive. Well, I'm a kingpin.
In my teen years and in my college days, I was the dork, the one that studied really hard, got good grades, because I was the first in my family to go to college. I had a lot to live up to with that. So I had to make sure that while friends were out partying, their grades were C's, I needed to get A's, and I knew it. Knowing what I know now of what life had in store for me, maybe I would have had a little bit more fun. I probably would have.
We started her on chemotherapy, and that chemotherapy really didn't work for her. So we tried another kind of chemotherapy.
I know that chemotherapy is supposed to kill cancer, it can give you back your life. Chemotherapy made me feel like I had cancer. I felt like death warmed over. I got severe peripheral neuropathy from the chemotherapy. That's numbness and tingling in your hands and feet. It wasn't just hands and feet though, it went up my leg and I couldn't hold a pencil to write. Your skin peels off, all of it, peels off. So it looks like you have a massive sunburn everywhere. I was itchy everywhere. It was like an itch that you could never stop.
And that chemotherapy didn't work for her. And then I sent her to get high-dose interleukin-2.
My chest started to fill up with fluid. I remember sitting across just like this from my mom in the family room of her house, and I would have to go, Mom, what are we going to do? Dude, that was my breathing.
And the high-dose interleukin-2 didn't work.
At the same time, though, I had an MRI done, which I had not had done previously, and they found a brain tumor.
So she needed radiation to her brain.
Do you know when you break your neck and that thing sits on your shoulders and they screw it into your head? That's what they put on you when you have to be bolted to a table when they give you brain radiation. So I had somebody come at me with a drill and screw things into my head while fully awake and conscious.
The life expectancy of someone with metastatic melanoma at the time was about 7 months. So we are way into that at this point.
We published the paper in 1996. Everybody kind of went, wow. And that was kind of it.
We were all very skeptical of the science of cancer immunology.
I wasn't sure that what he had done successfully in a mouse, in a very controlled setting, would ultimately translate to a method that would work in a human being.
You're going to be hearing a good deal more about something called interferon these days.
Skepticism about an immunotherapy drug for cancer stems from the 1980s.
Researchers are now calling a new cancer treatment the most promising in 30 years.
When attempts at using the immune system to fight cancer offered enormous promise.
With a drug called interleukin-2.
And then, devastating disappointment.
Because fewer patients than expected seemed to benefit.
For years, there were clinical trials, and none of these worked.
We started to see some bad side effects.
And I can tell you these were not happy meetings to attend. They all started with, we did this experiment in animals in our lab, and it looked great, and then we did a clinical trial, and nobody got better. Years of that had led to a view that immunotherapy would just never work.
20 or 30 years ago, cancer was not a very big thing for drug companies. The drugs didn't work that well, the patients didn't live that long. The much more lucrative drugs were for heart disease or diabetes, chronic diseases that people would take drugs for the rest of their lives.
Jim first goes to BMS, who had held the patents, and Peter Lindsley worked for them at the time. The guys at BMS had told him, we like CTLA-4, but we think it has other applications. But in fact, they weren't doing anything clinically with it at all.
I was very disappointed in the initial response, but it got more and more frustrating with time. It was just no, place after place after place. The scientists would get happy, and excited, and attorneys would say, no, you can't do this because Bristol Myers Squibb already has filed for a patent on their antibody. And I said, have you read their patent? Their patent is backwards. But still, the company would say, well, BMS has got more lawyers than we do. They're a big company, and so, too bad.
So there were all kinds of things that stood in the way of this happening. And I would say, his experiment in this was to just keep fighting to say, there's something here, there's something here, there's something here.
And that's where I really give him credit. It's like a bloodhound on the scent. He's not going to get taken off the scent until he's found the fox.
Like I said, this model is out of hard to control because I'm injecting live tumor cells initially in terms of because the mice, they always have a tumor.
I would always plan vacations, places where he could not get a phone call and he could not be connected to the internet.
Dad is doing a great job of chopping wood. Robert, go over there and stand by Dad. Okay.
It was about 2 years that nobody would listen, and So it was getting depressing. My brother Mike had metastatic prostate cancer. He presented with a lot of joint pain, which is how they found it.
We often had Thanksgiving with Mike. For many years in Berkeley, he'd come out, and so I got to know him quite well. Mike got diagnosed in his 50s with metastatic prostate cancer to the bone. That's how he was first diagnosed, and that pretty much is a death sentence. And that also, during this time, while Jim was trying to march forward this new therapy, he really wanted to be able to give this to his brother.
I need to get it into people. I need to get it into patients.
Jim turns his search towards smaller and smaller biotech companies, looking for anyone willing to take a gamble on his idea. In Boulder, Colorado, he finds Alan Corman, who is creating drugs for autoimmune disease.
My interest was in immunology. I didn't really know much about cancer biology or cancer therapy. I think it was useful not to be biased by the current thinking of how to go after cancer.
The concept of jumping into a problem and being the naive one is really useful. The people that have been in that field very long and can doing the exact same thing, I think you get tunnel vision.
Corman's job will be to take the CTLA-4 antibody that Jim and Max developed for mice and make it into a drug that can work safely in humans. The drug will be called ipilimumab.
I ended up collaborating with someone that I knew from graduate school, a guy named Nils Lomberg.
Nils Lomberg works for Medarex, a small biotech company based in San Francisco.
I got a call from Alan. We started talking about the idea of ipilimumab. Ipilimumab hadn't been created yet, and I convinced him to let me in on this. I was fascinated.
Jim and Alan Corman and Nils Lomberg all get together at the bar in the basement of the Hotel Boulderado, right in downtown Boulder.
Jim's primary concern was that we move quickly. I think he was very skeptical of the whole biotech pharmaceutical industry. He didn't think we had the urgency. I ended up sitting up with him most of the night with a bottle of whiskey. Out of that conversation, we had a handshake agreement. And with that handshake, we started work back in California, back in the lab. 466 days later, we had ipilimumab in a patient. And I think that's a world's record.
Phase 1 tests relatively small number of people generally for safety. Sometimes they get a little hint of whether the drug's working, but generally it's just to make sure that the drug is not going to harm somebody.
We picked a dose that was, a coin toss that was based, somewhat loosely on what we had done in the mouse. But clearly, we didn't know enough about how to figure out its potency.
When we were developing the phase 1 clinical trials, there were many stops and starts. There were many times when it was wondered whether or not the clinical trial could even start. Which tumor type should we do it in? Should it be all comers? Should we focus on melanoma patients? How many patients should we enroll in the phase 1 clinical trial? How much of a budget did we have to enroll these patients? What was the starting dose of the drug that we were going to give? Were we taking the starting dose just based on mouse data, which by the way is what we did?
When drugs go into phase 1 trials, their first trials in people, 9 out of 10 or more fail. And that's it. You pull the plug at that first stage.
I ran that clinical trial at that time. It was amazing because 3 out of the 12 patients, was a very small clinical trial, 3 out of the 12 patients showed that all of the tumor went away. We never see this in a phase 1 trial. It's so rare that it's sort of like, finding the pot at the end of the rainbow. You feel like, am I really seeing this or am I imagining it, right? It's just unbelievable.
I became aware that CTLA-4 was the focus of an early set of clinical trials that was, through the clinical trial grapevine, producing a little bit of activity in patients with advanced melanoma, and obviously I wanted to be part of that.
I remember going Christmas shopping in a wheelchair in the mall where we grew up. That was probably the worst Christmas ever. I remember sitting underneath the Christmas tree with Rob, and I was a huddling mess on the floor. And he just held me and told me to work it out. That Christmas was hard because I didn't think I was going to see the next one.
Comes to clinic and I say, I think that we're really at the point where the readily available standard options are not working for you. There is a clinical trial that you could consider participating in.
I remember Dr. Walchuk explaining it to me in very, very simple terms, that instead of an outside agent like chemotherapy, your body then uses its own immune system to fight off cancer. It allows it to turn on the immune system and recognize cancer in a way that it previously couldn't.
Sharon got 4 doses of ipilimumab.
The treatments were not like chemotherapy. Chemotherapy, you go back 3 or 4 days in that week, you live at that oncologist office, it's your home. This was different. I had a shot in my leg, and then I had a 90-minute IV infusion. That was it. Because I wasn't in chemotherapy either, I didn't feel nauseous. I didn't feel the incredible fatigue that came along with it. I noticed a difference in myself just from changing treatments.
Once a drug is proven safe in phase 1 trials, it moves into phase 2 and phase 3. These trials include far more patients studied over much longer time periods. They can cost hundreds of millions of dollars.
We never could have developed ipilimumab as a small company, even a publicly traded company. We never could have raised that much money.
Medarex has to find a partner with deep pockets, but the big pharma companies are wary of the risk. For any hope of success, ipilimumab needs a champion inside the pharmaceutical industry.
When I left the NIH to go to industry, I went to Bayer that was building an oncology program. I really wanted to supervise the development of IPI. When Bayer declined, I left Bayer after 7 years, specifically to bring ipilimumab forward. BMS was not sure they were going to take it at all, so I came at just the right time. And I was ready to advocate for it. I went to BMS. I said, let me walk you through why this is important.
I must admit I was apprehensive. The fact that BMS had another antibody would have been one of the reasons that a lot of companies had given for not working with us. BMS can kill you.
Stocks to watch today. A big one will be Bristol-Myers Squibb.
There were other reasons to be apprehensive of Bristol-Myers Squibb.
Bristol-Myers had a pretty good reputation. Then that started to fade. They did a deal for a drug from a small company called Imclone.
The drug sparked an insider trading scandal that led to a 7-year prison sentence.
Pessimism propelled by news that yet another corporate giant may have accounting problems. Today, it's Bristol-Myers Squibb in the government's crosshairs.
So they went through in the early to mid-2000s a very hard period
Bristol Myers Squibb, one of the leaders in the industry, forecast disappointing earnings growth for its second quarter.
So what we were asking BMS to do is invest a healthy amount of money into the belief that these drugs were going to work.
It was against the grain. A lot of our external advisors said we were probably investing too much in immunotherapy because nothing had worked before.
My presentation focused not on the response rate, which was very low at the time, but one patient who had received, I think it was a single dose at the time, that now 2 and 3 years out was still in remission.
The very first public meeting, Rachel made it a point for me to come and sit next to her. And I'll never forget, because I was sitting there and there were some people that had very little understanding of immunology that were making comments and, you know, about what they thought this meant or that meant, everything. I was just sitting there going, oh, bullshit. And I was just sort of under my breath, you know, going, oh my God. And obviously getting agitated. And Rachel, you know, kept looking at me, and afterwards, she says, okay, well, I wrote down every time you started snorting and stuff, and so what about this? What's wrong with that? What's wrong with this, from her notes? And so we went over the notes for a while. It was really a lot of fun.
I was part of the group of people who was following, let's call it the conventional path of cancer research. Jim Allison and the immunology crowd was thinking about it differently, and the mainstream was more or less ignoring that.
Innovation is extremely difficult. There was somebody, a regulatory person on my team, who yelled at me, screamed at me. We don't get it. We're wasting our time. And it was not just the members of the team, because back in the day, most of the doctors were not immuno-oncology believers. That's why it was hard to get patients on the study initially. That's why it took so long. There was like a year where that clinical study could not be enrolled. It was very, very slow.
No one wanted to hear about another cancer immunotherapy agent. It was, you know, forget it, we don't want to talk to you. And you can imagine a lot of doors slammed in his face.
You might argue that there is a defense mechanism against new ideas. They are seen as mutations, as cancerous to a body of thought. You don't know whether the new idea is something potent or deadly. And that's why they're often viewed with skepticism, and that's why it takes a really powerful idea combined with someone willing to push it forward to make it happen.
I inherited predisposition for cancer. My mother had melanoma, my grandfather had pancreatic cancer, and my beloved uncle got glioma when he was 58. So I know that cancer is coming for me. I know it's coming for my family. I'm just about the age my uncle was when he got his brain tumor. The stakes are so high. The opportunity is so great that somehow it just didn't matter when people yelled at me or created barriers. Because we're going to make it work. We're going to make it happen.
I'm still in Berkeley, but the action had moved to New York, where the companies are, and a lot of the studies were going on at Sloan Kettering. I thought, well, there's some not so good stuff happening to my baby over there, and I better move again, you know.
Jim decides he needs to be in New York where the major trials are going on. When I asked Jim why he needed to be there, he said, I went there to be a nuisance. He wanted to stay on the scientists, he wanted to stay on the FDA, he wanted to stay on Bristol Myers Squibb.
Anytime a lab moves, it's always a tough decision, especially if you're moving, you know, across the country. People have roots in a given area. I'd been in the Bay Area for, you know, almost a dozen years at that point. So when Jim decided to move from Berkeley to Sloan Kettering, I think it's a testament to how dedicated all of us were to this work and the promise we saw in it, that I would say at least 90% of the people in Jim's lab picked up and moved cross country. It was a tough move in that, you know, you're moving all your stuff, you're moving a lab, things in the labs are delicate. We actually lost an entire freezer full of sort of critical, unreplaceable reagents. Somewhere in the desert, halfway across, the refrigerated truck that it was plugged into blew a fuse. So there was some pain to go with that move.
Jim being in New York was truly a fish out of water. I'm not sure Jim walked more than 3 blocks from his apartment on the Upper East Side. But, you know, he didn't move to New York to take advantage of what New York has to offer. He moved to take advantage of what Sloan Kettering had to offer.
Putting pressure on Jim is Pfizer, the world's largest drug company. Seeing the progress of IPI, Pfizer has launched a competitive cancer drug, drawing from Jeffrey Bluestone's work.
I went to Pfizer, and they got excited about it. We were on track. I was working on them to make the drug, and things looked really good.
Tremelimumab was being developed by Pfizer, and the early signals for both of the medicines were remarkably similar.
Pfizer came in late when they saw reasons to believe. Very classic. We think maybe you have something here. We have one just like it. We're going to beat you.
The race is on, and watching it closely is the FDA, the U.S. government agency that determines whether a drug is safe and effective enough for patients. The FDA is judging the clinical trials by standards developed for evaluating chemotherapy.
The rules said if a patient hadn't had their tumor shrink by 30% within 12 weeks, they failed.
In Pfizer's trial, the tumors don't shrink after 12 weeks. They grow. Failing to meet FDA standards, Pfizer decides to cut its losses.
Pfizer did this giant trial, and just everybody was watching. It failed. You know, a big blow. You know, the biggest drug company in the world just abandons the whole field. I mean, everybody, when we started out, one of the main criticisms we had is immunotherapy's never worked, it'll never work, it's a bunch of voodoo, and you shouldn't even try it. Now here's this big failure.
BMS thought, there's only one other in the class. It just failed. We don't want to put more money in. In fact, should we be closing the studies you're doing now?
Many of my colleagues said, this will never work. This is a dry well.
Jim struggles to make it clear that ipilimumab is not a chemotherapy drug and doesn't behave like one.
People go back to their community. As soon as they got back, they'd put them on some chemotherapy drugs that had been shown never to work. But now they're responding to such and such, and said, yeah, right. You know, there's another explanation. That's that they're just responding late, which is what I showed you in the mice.
You go back to Jim's early experiments in mice with CTLA-4. Jim observed that the T cells were entering into the tumors and they were expanding in size before the T cells then were able to destroy the tumors, and then they would shrink in size. So Jim had to convince Bristol Myers Squibb to do a much more expensive, much more lengthy trial.
Unlike Jim, who, to his credit, persevered and did whatever it took, I felt I had done exactly what I normally would do, which is to just let the other folks make the drug. Little did I know at the time that they would give up on it so quickly.
Jim was visibly angry. He'd just go into a tirade. I've got to make this happen, because you'll never get fired for saying no if you're responsible for spending a lot of money.
The skepticism was widespread, and frankly, Jim experienced it probably every day.
Every time I'd see him, you could see his blood pressure going up. That was hard on him during those years, and hard on people around him.
That was a trying time. Jim became rather obsessed. The science and the project just consumed him, and I really couldn't be a part of that. With no science background of my own, our son off in college, we had less and less in common. That didn't mean that we didn't admire each other and care greatly for each other, but we just decided to go our separate ways.
I was afraid this drug was going to go down, and I invested not just myself, but my family. Yeah, it was a tough time. I think any great, big, disruptive change that you're pushing for is going to take some single-mindedness. Whether it has to be sacrificed, I don't know. I suppose maybe it does.
At Memorial Sloan Kettering Cancer Center, doctors are seeing in patients exactly what Jim has predicted.
Patients were coming in and telling us that they felt well. Even though the CAT scans are showing that the tumors are growing, these patients said, I'm feeling really great on this treatment.
Some people, after they came off of the clinical trials of ipilimumab, began to get better with no further therapy. And this was quite striking.
And so at that point, clinicians like Jed Walchuk asked these kind of immovable, implacable forces, Big Pharma and the United States government, the FDA, to change the trial.
The risk BMS was taking at the time was we were committing to years more of clinical study before we'd ever get approval. In an environment where there was tremendous uncertainty about whether this thing was going to work or not.
We were asked to do another 5 years of expensive, long, tedious trials. There were investors that didn't think this was going to work. We hadn't had a long track record of recent success. This takes a toll. This takes a toll.
My brother was in the final throes of metastatic prostate cancer, and the drug was in trials by then, and there were really good results coming back. But it was just not possible, and that was, it was frustrating. But at the time, I just wasn't so sure about whether it would really, at the end of the day, do my brother any good, because he was so sick by then.
I remember him calling me from Dallas when, Mike was dying, and he was in his arms at the time. And you can imagine, what that would do to you.
When I was first told I had prostate cancer, I was terrified because it was a week after my brother had died. I'd been working with the prostate group at Sloan Kettering. So I went over to the oncologist's office. The secretary said he's with somebody. I said, I don't care. Beat on his door. And said, can I talk with you? And he said, I said, no, now. I need to talk to you right now. My oncologist said, in your case, because of family history, I think we better take it out. Anyway, they caught it very early.
Sharon came back for her first set of imaging. I got a phone call from the radiologist. Now, usually, you should know, radiologists don't call with good news. They call to make you aware of some awfulness that they have seen on a scan. And this was a very serious radiologist, very well respected. And he called and he said, Jed, I'm reading your patient Sharon's CAT scans. And he goes, I have to ask you something. He goes, what happened to her? I'm like, what do you mean what happened? He goes, what did she get? All the cancer went away. And so that was an important moment for many of us.
With enough patients showing improved health, BMS decides to commit to the full 5-year study and accept the enormous financial risk.
Bristol Myers Squibb made the decision with everybody in the room then to use overall survival as the endpoint and not to use the typical sort of tumor shrinkage immediately endpoint, but look to see how patients did over a long period of time.
I give the team at Bristol Myers, and Rachel was the lead of that team, credit for listening to us and for being open to the observations that we were really making on the front lines and to working with us to find a way to capture this new biology.
Most companies and most people in those companies at that time would wash their hands and walk away. And so I give great credit to BMS. They went with the science. They believed the scientist, and they believed the data.
Dr. Walchuk, in his ever so calm voice, said, so Sharon, the man that invented this is here today. Would you like to meet him? Yes!
He calls me up, says, come down. He didn't tell me that. I found out why I was there only when I walked into the room.
Dr. Allison walked in, and I pretty much almost lifted him up off the floor, gave him a giant bear hug. I just couldn't talk. I couldn't talk. How in the world are you supposed to adequately thank somebody standing across from you that you are 100% positive that without them, you wouldn't be here? There is no thank you for that.
Oh, meeting her was amazing because until then, it was just numbers. It was a concept. She was the first patient I'd ever met.
And all we could think about at the end of that was, we got to get a picture. We look around the room, and nobody has a camera. The only camera that we could find was one that they took pictures of a melanoma with to send out and get studied. It's not the best quality in the world, but it definitely memorializes one of the best days ever. Every single scan since, I have another MRI coming up here in about 2 weeks, so say a little prayer. No evidence of disease. In every one since.
Now she has her family, 2 kids, 2 beautiful kids. I still choke up when I think about that.
In 2011, 15 years after Jim Allison made his initial discovery, the clinical trials are finally complete.
I remember the moment when we saw the data. I can see the people around me. It is imprinted like a picture. Because we knew we were changing the world.
Rachel Humphrey came running up the stairs, breathless, to show me the survival curves, one for the control and one for the patients that received IPI. One curve stayed different for as long as we studied. After years, you start to think that maybe you've cured these patients.
I happened to be at a melanoma meeting at Cold Spring Harbor, sitting next to Harold Varmus, and I had my iPad on and kept checking it. And he said, what are you doing? And I said, well, the FDA is supposed to announce its decision on IPI today. All of a sudden, I got this email. It was about 11 on the East Coast, which meant it was 8 in California. And there's Alan in the Metarex crowd holding glasses of whiskey. And I looked at Harold, I said, he said, I guess that's good news. I said, I think that's good news, yes. And he said, either that or they just like to drink in the morning.
And others like him have only begun the thing. Each generation will build on the next. We're not nearly at the end of this story. I mean, we're somewhere in the very first few chapters.
This happens fairly regularly. I get emails, but many times a handwritten note. This is from somebody I've never met. Dear Dr. Allison, 2 years ago today, my 63-year young husband, Richard, was diagnosed with metastatic melanoma to the brain and everywhere else. Thanks to you, your fortitude and determination to prove that immunotherapy had promise, Richard is alive and well, healthy and happy, and has been treatment-free for over a year. Anyway, it says, the words thank you do not begin to cover the gratitude we have for you and your persistence through all this to see that immunotherapy be brought to people worldwide. Our family is eternally grateful for the extra years we've given us with Richard, a wonderful father, husband. Sincerely, it's from his wife. For sure, it's knowing that we've done something like this what makes it all worthwhile. So, I need a drink.
Roll me up and smoke me when I die. And if anyone don't like it, look up in the eye. I didn't come here and I ain't leaving, so don't sit around and cry. Just roll me up and smoke me when I die. Now you won't see no sad and teary-eyed when I get my wings and it's my time to fly. Just call my friends and tell them there's a party, come on by. And just roll me up and smoke me when I die.
When I played with Willie Nelson at the Austin City Limits Festival, I was scared.
Look, I'm in ya.
It was hard to relax, when I'm playing with my buddies, it's like fun. But when you're playing with Willie, I gotta get this, I gotta get this. thank you so much. I'm so honored. You know, of course, it's a scientist's dream.
We think that the discoveries that you have made has enabled an entirely new principle for cancer therapy. I mean, we think it's absolutely fantastic.
Me out and build a roaring fire. Throw me in the flames for about an hour. Take me out and twist me up and point me toward the sky. And just roll me up and smoke me when I die. Roll me up and smoke me when I die. And if anyone don't like it, look 'em in the eye. I didn't come here and I ain't leaving, so don't sit around and cry. Just roll me up and smoke me when I die. I didn't come here and I ain't leaving, so don't sit around and cry. Just when I die.
Pam Sharma was tenured at MD Anderson. She was a big name in the cancer world in her own right. It was talking professionally for quite a few years. And then eventually there was a romance. Jim said to her one day, "There's no one who can stand either of us. You know, who else is going to talk about T cells all day? You know, we should get married."
¿Qué tal? Emocionante, ¿no? De verdad, una experiencia increíble el haber vivido en toda la plática, en todas las preguntas y en toda esta historia de vida que nos ha permitido y compartido el doctor Jim Allison. Por hoy ha sido pues prácticamente todo y los esperamos mañana, ya al inicio de las sesiones académicas, desde muy temprano, a las 7:30 de la mañana, con simposiums de cáncer renal metastásico, cáncer de mama, tratamiento personalizado en cáncer de mama, carcinoma ureterial metastásico, carcinoma de pulmón y hepatocelular. Todos ellos desde las 7:30, 8:30, nuestros simposios satélites, y los esperamos porque tenemos una agenda sumamente interesante. Simposios con la Sociedad de Cirugía Oncológica de Estados Unidos, la Sociedad Europea de Cirugía Oncológica de Estados Unidos y las académicas con inmunoterapia. También vamos a tener un canal de cáncer infantil con los simposios de SMeO AMOP, Oncología Pediátrica y toda una línea de investigación y de conocimientos nuevos en oncología pediátrica. Vamos a tener también los simposios de cabeza y cuello, cáncer ginecológico, cáncer colorectal, trabajos libres, sarcomas, genética en cáncer, mujeres en oncología y la presentación de trabajos libres, así como también un segundo bloque de cáncer ginecológico. Nos vemos mañana, los esperamos mañana. Muchas gracias.